In this video, I want to talk a bit about the accessory organs of digestion. We talked about some accessory organs and structures when we discussed the mouth. We talked about the salivary glands there. In this video, I want to focus on the accessory organs that release their substances into the small intestine, which means that I'm talking about the liver, the gallbladder, and the pancreas. Here's a cadaver view to see a bit more what these organs actually look like. You've got the liver here, the gallbladder kind of tucked up underneath the liver, and the pancreas posterior to the large intestine and kind of tucked into the curve of the duodenum. In this image and this image here, the bulk of the liver has been cut away, and you've also had the gallbladder opened up. With this view, and this is a little more zoomed in, you can also see the structures, the tubes that carry the products of the liver and the pancreas to the small intestine. You've got the bile duct here, and the pancreatic duct, which runs along the side of the pancreas, picking up pancreatic juices, and both of them empty into the duodenum at the hepatopancreatic ampulla. Let's talk about the liver first. The liver is the largest gland in the body. It's pretty enormous relative to body size. It's composed of these structural units called lobules, which are roughly hexagonal. Honestly, you're never going to see a microscopic image where you can see the lobules this clearly. This is about the best you can expect to see, where you can see a little bit of connective tissue separating this lobule off. I've highlighted a lobule here so that you can see where individual lobules are. Lobules are defined by a central vein with hepatocytes around it, and the hepatocytes sort of radiate out from the central vein. And at the corner of each lobule, you have a portal triad. Portal triad is composed of a bile duct, an arteriole, and a venule. This is sort of a cartoon version of a liver lobule that really illustrates the blood vessels and bile ducts that run through the lobule. Notice the portal triads. At each corner, there's one, there's one, there's one, there's one, there's one, and there's one. So basically a portal triad at each corner of the lobule. So we have the bile duct. And the bile duct, of course, drains bile from the liver. So bile that is actually made by the hepatocytes in here drains into the bile canaliculi, and then the bile canaliculi collect into the bile duct. You also have two blood vessels. You have a portal arteriole. The portal arteriole is bringing oxygen-rich blood from the heart to the liver. And then you have the portal venule. And the portal venule is bringing blood from the digestive viscera. So one of the important functions of the liver is to actually process nutrients coming from the digestive tract. So blood coming from the digestive tract that is rich with nutrients from the food that you've eaten that have been digested and absorbed, that blood is swept directly to the liver for processing. And the portal venules, there, they bring that nutrient-rich blood into the lobule. You also, apart from the hepatocytes, which are the basic building blocks of the liver and the functional cells of the liver, you have hepatic macrophages that help monitor that blood. They phagocytose damaged red blood cells, they phagocytose cell debris, any pathogens that are in that blood coming in. These hepatic macrophages are sometimes also called Kupffer cells. So you should be familiar with that term. Let's talk a little bit more about that portal circulation that brings blood from the digestive tract to the liver. A portal system, when you're talking about the circulatory system, a portal system is any arrangement of blood vessels where you have a capillary bed draining directly into another capillary bed. So, in the hepatic portal circulation, you have capillary beds in around the digestive tract, in the wall of the digestive tract, picking up nutrients. And the blood from those capillary beds are draining into a vein, into the hepatic portal vein, and carried directly to other capillary beds that run throughout the liver. So this nutrient-rich blood is going straight to the liver, and anything absorbed into the blood goes straight to the liver for processing. The hepatocytes of the liver have a number of important functions. Hepatocyte literally just means liver cell. So it should be fairly straightforward to remember that hepatocytes are the cells of the liver. I want to focus initially on what the hepatocytes do to blood, to the nutrients in blood coming from the digestive tract, and that's where these three functions come in. These three functions are why it's important that blood from the digestive tract goes straight to the liver. First off, because the liver is key in detoxification. So if you've taken in any harmless substances that have been absorbed, they are most likely broken down by the liver. Many drugs are processed by the liver and converted into harmless substances that can be excreted from the body. Alcohol is broken down in the liver as well. So that detoxification function is convenient. The liver also breaks down nitrogenous wastes in blood to urea that can be excreted by the kidneys. So the liver is actually important in urine formation because it makes the urea that's excreted. The liver also acts as a reservoir of fat-soluble vitamins. So if you are consuming vitamins A, D, E, or K in your diet, these are the fat-soluble vitamins, they are stored in the liver. So if your diet is deficient in these vitamins, you might want to consider adding some liver to your diet. Not human liver, of course. The liver also processes other nutrients that are coming in from the digestive tract. Glucose coming in from digested and absorbed carbohydrates can be stored in the liver as glycogen. And amino acids that have been absorbed are used by the liver to make various plasma proteins. The liver makes most of the proteins in the plasma, like albumin, and it needs lots of amino acids in order to do that. So it kind of gets first stab at taking those amino acids it needs. The fourth function of the liver is one that I want to talk a little bit more about, and that's the production of bile. This is where the liver actually helps digestion out. The liver doesn't produce digestive enzymes, but bile acts as an emulsifier. So let's talk a little bit more about that. Bile is this kind of yellowish-greenish solution, a little bit alkaline. It contains two main substances. It has the bile salts, and the bile salts are really the functional portion of the bile. The bile salts are the emulsifiers. They're made from cholesterol. So cholesterol does actually have functional uses in the body. And one of those uses is to make the bile salts so that your body can better break down fats. The bile salts are actually reabsorbed by the ileum and recycled. Soluble fiber in the diet kind of carries bile salts out, and that's why a diet that's high in soluble fiber can actually reduce your blood cholesterol levels a bit, because if that dietary fiber is carrying bile salts out of the body, preventing reabsorption, then your liver needs to use some more cholesterol from the blood in order to make new bile salts. And the other thing that you find in bile is bilirubin, and bilirubin is what gives bile its color. It's also what makes feces brown, and it's what builds up in the skin if you have liver issues, if your liver isn't functioning and you develop jaundice, it's what turns the skin yellow. There are a few other things in bile. There's some cholesterol in bile. There are other lipids, phospholipids, and triglycerides, and electrolytes, some ions in bile as well. But the bile salts are really the major functional part of bile, the emulsifiers. So what the bile salts do is when you have a large fat globule that is entering in your food, the digestive enzymes that need to act on that fat, that need to break apart the triglycerides in that fat, the digestive enzymes are in a watery solution, right? And we know that oil and water don't normally mix. So the digestive enzymes can really only access the outside of that fat globule. They can't get in to all those triglycerides that are hidden away in the center of that enormous fat globule. So the bile salts are both hydrophobic and hydrophilic. They have a polar region that has a charge that is perfectly happy to interact with water, and a nonpolar region that has no charge that is perfectly happy to interact with the fat. So these bile salts, they basically coat the fat, breaking that globule apart into smaller droplets. And now, with the digestive enzymes, they can access a lot more of the triglycerides in these fat droplets. So they coat the fat, separate it up into smaller droplets so that the fat can stay suspended as these small droplets in the watery solution that the enzymes are in. This is just a slightly different way of looking at the emulsification. I like showing this because it illustrates the fact that the enzymes are in a different solution from the fat. You know, oil and water don't mix. The fat is up here in the oily part. The enzymes are down here in the watery juices of the GI tract. Bile lets the two mix so that the enzymes can act. The gallbladder also has a role to play in getting bile to the digestive tract. The gallbladder doesn't actually produce bile, that's only done in the liver. But what the gallbladder does is it stores and concentrates bile. So it acts like a little bile sac, holding that bile until it's needed by the small intestine. While bile is sitting in the gallbladder, water is getting drawn out of that bile, and that makes the bile more concentrated and more effective. The gallbladder can be removed if necessary, and the bile duct can take over an extent of bile storage, although it doesn't really concentrate the bile the same way. So some people who have their gallbladder removed may need to be careful about how much fat they eat in their diet. It really varies. Some people never notice a difference. Some people need to start watching what they eat. The reason why you might want a gallbladder removed is because if there's too much cholesterol in the bile, or if there are too few bile salts, you can get cholesterol crystallizing in the bile, forming gallstones. Like this one. The woman who had this gallstone, before she had it out, said that it felt like she was sleeping on a rock. And she was pretty much right. Our third accessory organ that releases juices to the small intestine is the pancreas. The pancreas produces digestive enzymes that break down all types of molecules in food. They can break down lipids, they can break down carbohydrates, and they can break down proteins. The pancreas also produces bicarbonate, so there's bicarbonate in the pancreatic juice, which helps neutralize the acid that's coming in from the stomach. So remember the stomach is highly acid. The chyme that's coming into the small intestine from the stomach is still pretty acidic. The bicarbonate from the pancreas helps neutralize some of that acidity. And this is important because the enzymes in the pancreatic juice and the enzymes at the brush border of the small intestine, they need a non-acidic pH in order to act. They would be deactivated by an acidic pH. As far as what enzymes the pancreas produces, there are enzymes that break down carbohydrates, like pancreatic amylase. So you have a salivary amylase that's released in the saliva, and you have a pancreatic amylase that's released by the pancreas. You also have proteins that break down, enzymes that break down proteins. You have trypsin, chymotrypsin, and carboxypeptidase. And for attacking the fats, you have the pancreatic lipases. Lipase is an enzyme that breaks down lipids. The pancreas also has a big job in helping our body use the nutrients. Remember, from the endocrine system, it produces insulin that tells our cells to take in glucose out of the blood and use it. The pancreas also produces glucagon, which, when we need more glucose in our blood, tells our cells to release glucose into the blood. So the pancreas has important endocrine functions as well. For this unit, for focusing on the digestive system, we want to focus on the exocrine functions, the production of the digestive enzymes and bicarbonate. This is a histological look at the pancreas, both in cartoon form and actual histology form, where you can see just the edge of a pancreatic islet up here, but remember, for the exocrine function, we're focusing on the acinar cells. And if you could kind of see the cells well defined, you would see these kind of clusters of acinar cells with granules at the surface. A zymogen is an enzyme precursor. So the pancreatic enzymes are actually released from the acinar cells as zymogens, these inactive forms. And then when these zymogens pass through the pancreatic duct and get into the small intestine, into the duodenum, that's when those zymogens are activated into the pancreatic enzymes. There are a number of hormones released by both the stomach and duodenum, and they have a role in controlling motility through the digestive tract, controlling secretion of products, including secretion of bile from the gallbladder and secretion of pancreatic enzymes. Gastrin is a stomach hormone. It acts primarily on the stomach. GIP is gastric inhibitory peptide, which actually promotes the endocrine function of the pancreas. It promotes the release of insulin and has an inhibitory effect on gastrin, as you might guess from the name. GIP is triggered by chyme entering the duodenum. VIP, vasoactive intestinal peptide, is also triggered by chyme entering into the duodenum. And VIP, vasoactive, so you know it acts on the blood vessels. What it actually does is it causes the intestinal capillaries to dilate. So you get more blood flowing through those intestinal capillaries, ready to pick up those nutrients that are going to be absorbed from the small intestine. So it's kind of nifty, actually. You know, just as you get food, both chyme, entering into the small intestine, ready to have digestion completed and ready for its nutrients to be absorbed, you get more blood flowing through the capillaries, ready to receive those nutrients. Secretin and CCK are the ones that really act on the accessory organs. Secretin and CCK both trigger the release of pancreatic enzymes, and they trigger secretion of bile, and they trigger contraction of the gallbladder, so that the gallbladder squeezes and ejects some of the stored and concentrated bile into the duodenum. CCK stands for cholecystokinin. All of these hormones act to maximize the efficiency of nutrient absorption and get those nutrients into the tissues. Okay, after spending some time with this video and studying it, you should be able to talk about the digestive functions of the accessory organs. You should also be able to talk about the non-digestive functions of the liver, so that processing of blood-borne nutrients, and a little bit about the non-digestive functions of the pancreas, which is basically a review from the endocrine system. You should be able to talk about the importance of the hepatic portal circulation. You should be able to talk a fair amount, fairly extensively, about bile, what it does, how it does it. And you should be able to talk about these hormones: gastric inhibitory peptide, vasoactive intestinal peptide, and cholecystokinin.